Good afternoon. I'm Shin Kwang-soo, Principal Researcher at the Salad Research Center, and I would like to deliver my appreciation for giving me an opportunity to present here. Thank you, BioInvest Fair staff members. Today I would like to cover this ADCLD COVID-19, the vaccine that we're currently working on developing, and I will deliver details. This is the status quo of development, and Dr. Yu has covered this well, so I'll just skip on this slide. And we are currently working on adenoviral vector to work on vaccination, and that you can refer to page 128. So I'll briefly introduce our vaccine. These adenoviral vector, which makes people catch cold, and we make use of this. And if we get rid of this part, the circle part, and then this virus cannot activate cold anymore in the human body. And to this part, we use this spike gene here, and we switch that part into 85 and 35 hybrid vector. And the efficacy followed by this is two. One is that we introduce this NOP gene, so we can effectively create an anti-gene body here, antigen. And if we make use of this virus O type, then there are some worries that if we use this in vivo, then there will be maybe some counterparts. So we can mitigate that risk, and we came up with this vector, and we made this SARS-CoV-2 spike gene as an antigen for the vaccine. And if we inject this spike gene, then we can come up with the vaccine. And I'll move on to the mechanism of action. And like I said, this vaccine uses this type 35. So when observed in the human body, then it can be adopted to this part. And if you adopt this, then within the cell, this vaccine antigen in the vector can create this immune system, and the protein created here that will work as a catalyst to prevent the infection and activate the T cell. And even if the human body contracts this virus, you can preemptively kill this virus. And the strength of this adenoviral vector is that you can kill and effectively induce those desired efficacy. And on top of that, we make use of spike protein expression, and we have our unique antigen. And we conducted some research, and thanks to our technology, when we made use of this antigen within the cell, you can come up with this wild antigen. Compared to that, we can effectively come up with antigen more successfully. And this, if you take a look at this slide, in the animal's body, the combined vaccine, the antigen, showed higher efficacy, so we can effectively work on this. So we named this ADCLD COVID-19 and kept working on research. And then I'll move on to the immunogenicity of ADCLD COVID-19. And through our tests on mice and monkeys, we saw this effective results. And we conducted T-cell analysis, and when we injected this vaccine, we saw that CTL, the vaccine, was well activated, and it killed the virus, and it was well activated to the antigen, and it absorbed to the body. And the T cell that is well activated can induce this antivirus, and also these T cells showed Th1 and Th2 responses, and there was significant good T1 activation. So we came up with Th1 reaction, and and we shouldn't induce Th2 activation. So we focused the T cell reaction, which is highly concentrated on Th1. And based on the research results, we conducted protective efficacy, and we used this transformed mouse strain. And after vaccination, and we exposed the animals to the COVID-19 and saw this reaction. And there were control groups of vaccine 1 and vaccine 2, and depending on the number of dosages, we saw if there are any significant differences. And as a result, the comparative group, after getting the dose, their weight kept decreasing, and when they died, those mice got vaccinated, recovered in the early stage, and none of the mice died. And despite if getting one single shot or getting twice, they showed this efficacy. And also, in terms of lung types, if we saw if there is any virus, and until treatment, the mice kept showing this virus. And if we vaccinated these mice from the third day, they didn't show any virus. And according to these research results, we saw that this mice experiment showed that our vaccination successfully decreases COVID-19. Next is our test on monkeys. We used the same measures on monkeys, and we induced this ELISA and neutralizing assay, and just like them, only with a single shot we showed this result. And the T cell reaction was well induced as well. And thanks after this successful immunization, after 7 weeks of treatment, we exposed this monkey to SARS-CoV-2 virus, and after this exposure, each day we collected this sample and we analyzed this, and those monkeys till the 4th week, those not vaccinated monkeys were kept getting virus. However, those vaccinated monkeys didn't show any virus. And even the lung samples, those vaccinated monkeys, all of them did not show any virus. So at the animal experiment level, we came up with the result that it is meaningful. And we conducted phase 1 and phase 2A, and we got authorization approval, and we are currently working on further development, and currently we are working on 2A phase, and we have completed 2A, and currently we're working on collecting samples, and if we complete that process, and we will be able to move on to the next stage. So today I will elaborate the additional analysis on phase 1. And for our phase 1 study, we categorized into 3 different dose levels, and we conducted this research in a timely measure, and we worked on 3 different groups: low dose, mid dose, and high dose. And we monitored those safety measures, and we worked on neutralizing antibody and T cell reaction, and we are currently working on this. So today I'll cover safety measures and post-vaccination neutralizing AB. So first of all, let's begin with safety measures, and it shows a lot of safety level. And if you take a look at the side effects, there were some levels. However, if you take a look at the grade 3, that there was no different challenging moments, and there were only 2 or 3 cases that showed side effects. And the most commonly side effects is just pain or tenderness. And this is something that commonly is monitored when vaccinated. So based on our judgment, our vaccination does not show any serious side effects compared to other vaccines. Next is immunogenicity. And if you take a look at the antibody binding AB titer, based on all the 30 samples, you can see that the combined antigen were detected here, and it increases accordingly. However, if you take a look at this mid dose and high dose, on the fourth week, there is less difference, and that is because there were only 10 samples from each group, and if we secure all the results of the 2A phases, then we will come up with further detailed information. And here is anti-RBD binding AB titer, and from all these samples, we saw this significant change. And just like spike gene, accord and it increased in accordance with the dose. However, since there were only 10 samples for each group, so we need more samples. And this slide shows SARS-CoV-2 NAB. And likewise, just like the previous slide, we saw this significant improvement, and we have successfully induced this. And in terms of math, and there were 200 from low dose and 427 from the mid dose group and 490 from the high dose group. And as you can see from the slide, if you compare the mid dose group and high dose group, there is no significant difference. So after the two-way phase analysis, we will come up with further details and come up with math. So in summary, the vaccine that we use it uses adenoviral vector, and it transforms the AD35 NOP vector, and the antigen uses our unique patent item to work on it. And our vaccine showed T-cell reaction and nitrogen reaction based on both mice and monkeys. And from both animal groups, it significantly prevents the virus. And so far, we're currently working on analyzing phase 1 and phase 2A, and the interim results of phase 1 tell us that 100% from all the samples showed that the vaccine neutralizing results were shown. And next is our plans in the future. And as I said before, we will soon complete the two-way phase process and analyze it. And we will soon begin
네.
our process development and process validation. And there will be some challenges collecting samples, so we are currently working on collecting more samples from other groups as well. So thank you for listening.